Hello class, this is my science class, 6th grade science class. Today we're going to be doing an investigation of gene copies, looking at traits and reproductions. This is our scholar, Cassandra. So we're going to briefly do our do now. So for the directions for our do now, you will have 4 minutes to accurately match these words with the corresponding definitions after we discuss key vocabulary words. We're not gonna go over each one of them just due to time. We're gonna go over the ones that students struggle with the most. So I'm gonna circulate the room, see which ones you guys need the most help with, and go over those. So I'm starting the 4-minute timer at level 0. You should be working on this independently and silently. So my 4-minute timer starts now, and you are matching these words with the corresponding definitions at a level 0. I see Marcus is doing it silently, level 0. I see Cassandra has started doing it silently at level 0. Great job. Great job. Oh, the 4-minute timer is up. Okay, so put your pencils down, dragging forward at the board. Please, students, bear with me. Thank you. All right. So one that I noticed you guys struggled with was number 1. So a characteristic that all members of a species have. So I noticed a few different answers. I noticed that some of you put future and some of you put trait. So a characteristic that all species have, I'm going to call on Cassandra.
So, all species will have a feature. I know that features are different from traits because features is something that all of them have, but traits are more specific.
Okay, so feature, can you give me an example of a feature? Because sometimes I think it helps when we use examples of what a feature might be.
Example of a feature of a bird would be all birds have beaks, all birds have wings, all birds have...
Okay, you can stop there. That'll be enough. So then if a feature is a characteristic that all members of a species have, then which number matches with a trait? Because I saw a lot of us get that confused. I'm going to call on Hector.
Hector. 11 would be a trait. Okay. A specific characteristic of an individual organism. Yeah, it's more specific than a feature. So I know, yeah, it'd be 11.
Okay, 11. And I'm going to give you an example of number 11. A specific characteristic of an individual organism. For example, that's a trait. A feature in a human would be that all humans have eyes. But a trait in a human would be that some humans have eye color that is blue, some humans have eye color that is red. That's a bad example. Some humans have eye color that's brown, some humans have blue, some humans have green. You guys know different eye colors. We all have eyes, just different colors. So a feature is what we all have. A trait is a specific characteristic in that species or individual. Good. And one more thing I wanted to go over that I saw students were struggling with was the difference between a structure and a function. We'll start off with function because it's the broader one. Which one of these numbers associates with function? I'm going to call on Bob. Bob, which one of these associates with function?
Number 2, how something works.
Okay, yeah. Number 2, how something works. To give you an example, actually, before I even give an example, I'm going to stamp, so if number 2 is function, then number 9 is structure. So 2 is function, 9 is structure. Go and fill that out if you haven't already. To give an example of this is a bicycle wheel. A bicycle wheel, the function of the wheel is to move forward, because it's a bicycle wheel, so it has to roll and move. But the structure of the bicycle wheel is the way it is shaped. So it is a circular shape so that it can move forward. So the structure can sometimes help the function of in this case, the protein or the molecule. Alright, we're moving on. Oops, sorry.
Again. Take it twice.
Alright, so here's what our learning target is for today, and this is going to be our big takeaway of the day. Can I have a reader? Cassandra, you've been participating a lot. I'll let you read this really quickly though.
Learning target. Students will be able to SWOBOT, gather evidence from digital models to summarize the effects of an organism's gene copies on the number of protein instructions provided for a cell.
Okay, so can someone put that in their own words really quickly? What will we be able to do by the end of this lesson?
We will be able to use the simulation, so the amplify sim, so that we can figure out why there's some people that have different proteins in their cell, and why there's some people who have more than one protein, and why some people have like 2 proteins. I know that not everybody has the same amount of proteins, and we try to figure out why that happens. I know it has something to do with reproduction, so yeah.
We'll leave it there. So great job with that. So we're gonna move on. Thank you for summarizing it. I'm gonna read it one more time. Students will be able to gather evidence from digital models to summarize the effects of organism gene copies on the number of protein instructions provided by the cell. That's a mouthful. But in order to be successful, I'm gonna breeze through these. Here's what we're gonna do. We're gonna circle back to these and our learning target at the end of the lesson to make sure that we did all of the things we said we were gonna do. So the first thing we're gonna do for our criteria for success, in order to be successful, we're gonna identify the number of proteins that are present in a cell, right? We're gonna go more into that in a second. We're gonna differentiate, sorry, I struggled with that word, between homozygous and heterozygous genotypes. Again, we're gonna make sure we know the difference between homozygous and heterozygous genotypes. And then we're going to explain why some individuals may have one type of protein in their body cell, while others have 2 types of protein in their cell. Key concepts: an organism has 2 copies of a gene for each feature. So this is just something that you're going to need to remember as we go throughout our lesson. An organism has 2 copies of a gene for each feature. Do you know where those 2 copies come from? Very quickly, stamp it.
Mommy and daddy tangled.
We'll leave it as they come from the parent of the organism.
You have 2 parents.
The parents, keyword 2.
When it comes to sexual reproduction, yeah, there's 2 parents. Mommy and daddy fell in love.
And I like how you stamped that when it comes to sexual reproduction, because when we know from a previous lesson, asexual reproduction
only takes 1.
So thank you for stamping that, so that we're very clear. We're looking at sexual reproduction with 2 parents. Okay, so today we will investigate the question: why do some organisms make 1 type of protein for a feature, and other organisms make 2? All right, so this good son. Bristol's protein example. Leo makes 2 types of proteins for Bristol's future. Where do we see the 2 types of protein? Looking at this diagram, hold up, someone's got to raise a silent hand. Where do we see 2 types of proteins in this diagram pictured below? I'm going to call on my scholar, Roger. Thank you, Roger.
Hey. In the cell, I see that there's different shapes, so I know there's different proteins. Like one of them is like really, a little like square-like, and then the other one's like rectangular. It's a little longer. And then I see when we look in the nucleus, we'll see R1, R2. And then whenever I see R1, I see that there's protein 1 over here, which is the really long rectangular one. And then when I see the R2, I see the square one. And protein 2 is made whenever I see R2 in the nucleus.
Okay, thank you.
Where the genes are found.
You answered all of it, but I'm gonna have someone else just add on just a little bit to the explanation. So what there's two ways to look at it. I can look at it, the protein gene in the nucleus, and we can see it says R1 and R2. And I can look at it, what's the other way that I can look at these two proteins? I can look at it, yeah, that works. I can look at it visually. I see that the two proteins have different shapes, representing two different proteins, right? So, in this case, we know that Leo makes two types of proteins for Bristol's futures, and yeah, we're gonna go on to this one. So, why do you think some organisms make one type of protein for the future while other organisms make two? So we're looking at these two diagrams. First things first, I want you guys to turn and talk to a partner really quickly. What do we notice about the proteins in the first spider versus the proteins in the second spider? And for the people in the back who can't see it, you can come up close to see it better. I'm just gonna zoom in on it so you guys can see it a little better. Turn and talk to your partner. You're gonna get I'd say about 30 seconds to turn and talk.
So I see that with Leo, he has R1, R2, but then when I look at Annie, I see that Annie has R1, R1. And so when I look inside of the cell for Annie, I only see the protein one. There's like the really long ones, the rectangular ones. But then when I look at Leo's cell, sometimes I'll see that there are the rectangular and the...
Just making sure to speak up. Sorry, so you're still in your turn and talk. Sorry about that. So we're gonna just wrap up the turn and talk, and I'm gonna have Christina stamp. What do we notice are the differences between these two spiders when we're looking really at the protein? So Christina is gonna stamp it nice and loud, Christina, so that all the people in the back can hear you.
So when I see Annie, before we didn't look at Annie, we only looked at Leo. So Leo had R1, R2, and so that's why I see protein 1 and protein 2. But then when I look at Annie, I see R1, R1, and I only see protein 1 in the cell. They look different.
Okay, so we noticed that they look different. This one's R1, R1, and this one's R2, R1, which means that there are two types of proteins in the second spider and one type of protein in the first spider. So now, why do you think, looking at the spiders, why do you think organisms make one type of protein for a future while other organisms make two? Looking at these spiders, what do we notice? Why might we really dive deep and investigate why some make one and some make two? I'm gonna call on Danny.
I think it has something to do with the parents, because we know all genes come from the parents. So maybe it has something to do with the parents.
Okay, so you think it has something to do with the parents? Okay, and you're on the right track. But what do we notice happens to the spiders themselves? When this spider has one protein and this spider has two proteins, what do we notice is any maybe physical differences or that we might have noticed from our previous lessons? Someone new, Michael.
So I noticed that it changes the trait, because when I look at those two proteins, when I see the R1s only, Annie has dense bristles, but then when I see the proteins 1 and 2 in Leo's, then he has sparse. It changes the traits, so it changes the phenotype, because that gene being in there changed the trait. When R1 and R1 genes were in there, and it's only protein 1 that's made, then Annie had dense bristles. But then when I see R1 and R2, and there are two proteins in there, that means that Leo has sparse traits, so his phenotype is a little different. He looks different with those different proteins.
Just want to have someone stamp that in their own words. Okay, thank you, Leah. Thank you, really appreciate you.
Yes, you go, Leah.
Yes, Leah. Just to be clear, we silently agree in this class. All right. So yes, the genotype determines the proteins, which will determine the phenotype. What does phenotype mean again? Because we're using some big vocab word from prior lessons. Just want to make sure we know what that means. Because I said it with the vocab words, which we should know at this point, because that's what we're gonna be using from here on out. But let's also make sure that we know what these words mean. So what does genotype mean and phenotype? Genotype is thinking about the word gene. Genotype is, I'm gonna call on someone, in the back, is that Fernando? Thank you, Fernando.
The genotype has to do with the genes.
Genotype means relating to the genes. Phenotype means,
I see the pH, so it has to do with the physical.
yeah, so the genotype determines, so the genes determine the proteins, which determine the physical traits of these two things. The genotype determines the proteins, which determines the phenotype. Okay? And I'm going to keep using those words as we go on throughout the school year. So we read this article yesterday. It was the second read of the hemophilia article. Okay. Gonna read this quick excerpt to you again, just so that's fresh in our mind. It says, I'm gonna read it off of here, and you can look up here or down at your papers. Does not matter. Just make sure you're at level 0 as I read this. You fall off your bike and you scrape your knee. Blood drips out, but soon a scab starts to form the bleeding. Oh wait, before I read this again, I'm gonna read this just so that we know what we're looking at and what we should be taking out. Sorry, I'm just gonna read this before I read this so you know what you should be looking out for. So we will now reread the part of hemophilia proteins and genes to help us understand why some organisms make one type of protein feature while other organisms make two. So that's what we're looking out for. Refreshing our memory on why some proteins from yesterday's reading make why some organisms make one protein and why some organisms make two proteins, all right? So I'm gonna read the reading, and it's just a paragraph, so it should only take about 2 minutes, tops. You fall off your bike and scrape your knee. Blood drips out, but soon a scab starts to form and the bleeding stops. Sounds simple, doesn't it? However, if you could zoom in to what's happening on the molecular scale, it's not simple at all. The blood includes 13 different protein molecules called clotting factors. When you injure yourself, these clotting factor proteins all connect and work together to form a solid clot, also known as a scab. If we're missing one of those 13 proteins, the clot wouldn't form, and the dangerous bleeding would continue. This is exactly what happens when a disease called hemophilia happens. People with hemophilia are missing one of the proteins needed for blood clotting. Alright, so rooting ourselves in this question. Why are some organisms make one type of protein for the future while other organisms make two types of protein for the future? Thinking about thinking about how this relates to hemophilia. Turn and talk to your partners for about a minute thinking about this question.
I have a question.
Before we go off to turn and talk, I'm going to take Cassandra's question. Cassandra, question?
Is hemophilia dangerous? It says it's dangerous, but who cares about the blood,
Okay, so hemophilia is basically if you get a cut on your body, there's different proteins, 13 to be exact, that form to clot your blood, meaning it stops you from bleeding. So is hemophilia dangerous? Yes, because think of it, if you didn't have, I don't want to tell you the answer to anything, but think of what would happen if your blood didn't stop flowing after you got a cut. It just kept on flowing and flowing and flowing. So simply put, yes, hemophilia is dangerous. I'm not going to go talk on too much about it because I don't want to answer the question itself for you. But think about, why do some organisms make one type of protein for the future while other organisms make two? Turn talk, 30 seconds. I'll actually give you guys a minute because it's a little tricky. 1 minute, go off and turn talk to your partner. Right now I see the whole left column is talking to the partner. I see the whole right side is talking to the column. Still waiting on the middle. 2 people in the middle are not talking. I'm going to walk over to the room. Okay. Good, now I see the whole class talking. All right, class, we're going to bring it back. and 5. We're gonna bring back in 4. We're gonna be back in 3. Eyes tracking forward at level 0 and 2. Your voices need to be off at a level 0, tracking forward and 1. Your voices are off. You're tracking forward and 0, and we are going to call on our scholar Francis right up here. Francis, so help us understand why some organisms make one type of protein for a future while other organisms make 2 in relationship to hemophilia. Francis, take it away.
Sometimes, I don't know, sometimes they're just, different people got different genes and they just feel, they're just off. Like their bodies just don't work right, you know? Like if you're supposed to have 13 and you're one who has 12, there's something wrong with you. Something, maybe the parents just sent you something bad, or maybe you just ate something bad, or maybe you just got bad genes, or.
Okay, we'll stop you right there, Francis. Thank you. So Francis is saying maybe something's just off when you're missing a protein. She might be right, but think about what does it say happens when you have hemophilia and you're missing a protein? What happens? What exactly happens?
You could die.
Someone give me a silent hand. Alrighty, I'm gonna recircle back to Francis. So,
hemophilia is dangerous. Like you could die out. Like you can't even put a towel. Like you just die. Like paper cut. You just dead.
So yeah, hemophilia allows blood clotting, and without it, you would just bleed out, and likely, you could die. I don't want to put that out there, but it's a real thing that happens in our life when a very small population of humans have hemophilia, and when they get cuts, sometimes they do unfortunately bleed out. So recircling back to like the core of this question, why do some organisms make one type of protein and some organisms make 2? Think about hemophilia. What happens when we don't have that protein in our body for blood clotting? Think about what happens. I'm gonna call on another scholar. I'm gonna call on Liz. Liz. Thank you, Liz. Take it away.
I don't know. I really don't understand this. I think I'm just gonna have to keep on reading it. Like I know there's somewhere in the text, like later, that talks about it. So I don't know.
Okay, so Liz doesn't know. All right, so to help you guys out, it says in the reading, when you injure yourself when blood clotting, factor proteins all connect and work together to form a solid clot, also known as a scab. If you are missing one of those, listen to this carefully because it says the answer. If you are missing one of those 13 proteins, the clotting wouldn't form and dangerous bleeding would continue. So what happens when you have hemophilia and some organisms make one type of protein for a future while other organisms make 2? What could happen if there's a different number of proteins? Gonna call on someone I haven't heard. Okay, I'll call on Devon.
You could die.
Yes, and how does that relate to the protein cell?
Because you're missing a protein.
Yeah, so sometimes when you're missing a protein, you're not genetically equipped to your environment or society around you. So the different number of proteins you have can even determine your survival, how you adapt to your surroundings. All right, moving on. Thank you for answering that, Devon. All right, so I'm gonna read this really quickly. I'm gonna read the first one, someone read the second one. Actually, someone can read the whole thing because this is all together. I'm gonna call on Mariama to read the whole thing.
You will reread the first paragraph of the cause of hemophilia. Yeah, I see that on my note catcher. As you read, pay particular attention to how the diagram and text fit together. Oh, I see that picture on my note catcher. True.
Oh, nice. So you will reread the first paragraph of causes of hemophilia section. As you read, we're going to be paying attention to the diagram and the text and how they fit together. And this is our note catcher that we're going to be paying attention to. I'm gonna hopefully think if you guys can see this. Alright, I'm gonna also.
I could read it out loud.
You think they could see it from me? Alright,
You get closer now.
I'm moving closer.
I'll read it. People with hemophilia are missing clotting factor proteins because of a problem with their genes. Different genes carry instruction for making different proteins. For every gene, there may be several possible versions. Different versions can also be called alleles. An organism has 2 copies of each gene. Those gene copies can either be the same, which is homozygous, or they can be different, which is heterozygous. If the gene copies are homozygous, there's only one type of protein produced, since both gene copies give the same instructions. If gene copies are heterozygous, there are 2 types of proteins, since each gene copy gives a different direction. So when I look at the picture, the homozygous, they both are pink, but then I look at the heterozygous and they're different colors. So that means they're different instructions. I see what they mean.
Yes. So, I love the pictures too. So the word homo, we'll use the easiest terms that you guys are most familiar with. There's homozygous and heterozygous, and there's homosexual and heterosexual. What do you think the word homo means when thinking about looking at the picture, the diagram? What do we notice when it's homozygous? And thinking about how that might relate to other words we've seen, like homosexual.
When I think of homo, they're the same color, and the homosexuals like their same sex. So like a man will like a man, and a girl will like a girl. But then the heterosexuals, they like people who look different from them. Like the girl ends up liking the boy who's different looking from her.
All right, so if we're to break down the word, homo is going to mean same. So if homo means same, hetero, like in heterozygous, that is going to mean what? Different, thank you. All right.
And so the questions?
I'm going to quickly zoom out. So looking back at page 2 of your notes and analyze Anne and Leo, so we're going to look at Anne's gene copies, and we're going to do this very quickly because I think we all have a really good gist of this at this point. So gene copies from Anne are what? You guys can...
When I look at Anne over here, I can zoom in. Annie is on over there. Their gene copies, I see R1 and R1. Is it homozygous or heterozygous? Yeah, so again, we said homo means the same. They have R1, R1. So are those the same or different? Answer that first. Same. So the same is going to be what? Homozygous.
Good. So homozygous for N. We're going to circle the homozygous for there. Leo, gene 2 copies.
Leo, I see, when I look at Leo cells, I see R1, R2.
To show you that clearly. Okay, thank you. So R1, R2, if they are different, they are heterozygous. Thank you.
So if Annie is homozygous, I'll just do this. Annie is homozygous and Leo is heterozygous. And so Annie, since she gets the same instruction, she only makes one type of protein. And then Leo, since he's heterozygous and he has 2 different gene copies, he makes 2 different proteins.
And that makes sense why? Looking at the words themselves.
Because mommy and daddy sent different instructions. So mommy sent one gene, one protein, and daddy sent another protein.
Good.
And then, this is the same question we keep on talking about.
Yeah, so again, we're going to recycle back to the question of the day. Why do some organisms make only one protein type for a feature and other organisms make 2?
It depends on what your parents sent you, because if your parents' genes code for one protein, code for the same protein, you're only going to have that one protein in your cell. But if you have parents who sent different proteins in your body, then you're going to have 2 different proteins in that cell. Some parents make the same protein and some parents make different proteins. Is that true?
Good, yes.
Should I
So
write
the
it down?
genotype, due to time, I'm gonna write it. You guys can copy it. So the genotype will determine the protein, which will determine our phenotype. Someone sum that up again. What does that mean in your own words? I'm gonna call on Mary Kate.
The genes determine what protein is in your body. And whatever protein's in your body is going to be expressed physically.
Good. All right. We're moving back toward our slides. Alrighty. So, homozygous, as we just went over, having gene versions that are the same. Heterozygous, meaning having gene versions that are different. So, in the pictures we observe, the organisms have either 2 copies of a gene that are the same version, or they have 2 different versions. Everybody call out. When I say go, you're going to tell me, when I say go, not yet, homo or heterozygous. 3, 2, 1, everybody go.
Homo.
Homozygous, because F1 and F1 are the same. This one, when I say
Hetero.
whoa, when I say go. 3, 2, 1, go.
Hetero.
Hetero, because
They're different.
they're different. Good job. All right, we can see homozygous, because they're the same. Heterozygous, because they're different. In the reading, we learned that scientists have specific terms to describe these combinations of gene versions. They can either be homozygous or heterozygous. All right. So again, we're recircling back to our investigation question. Why, this is going to be our takeaway. Why do some organisms make one type of protein for a future and other organisms make 2 types?
It's because the 2 copies can be the same, homozygous, and provide instructions for only one type of protein, and then, or they could be different and heterozygous and provide instructions for 2 types of proteins.
Good job. Before we end our lesson, we're just going to circle back to our criteria for our success. We obviously answered this one, our learning target. We gathered evidence from digital models to summarize the effects of organism gene copies on the number of protein instructions provided for a cell. But while doing that, did we identify the number of proteins present in a cell?
Yes.
Yes, we did. Did we Did we show differentiation between homozygous and heterozygous genotypes?
Yes.
Yes, we did that with the pictures. And did we explain why some individuals have one type of protein in their cell while others have 2 types of proteins in their cell?
Yes.
Okay. So we did all this. Bravo class, great class today. We're gonna be at level 0 and we're gonna start packing up.